And also, it had been around that time that Thomas Hunt Morgan, what did he do? Through flies?
Sex-linked inheritance.
All these thoughts are correct. In summary, he came up with the idea that chromosomes are the units of inheritance. They are the units that you get from your parents. And it was also known during that time that chromosomes themselves are made of two items. Actually, they're made of one item called chromatin, and chromatin is made of two items. What are those two items that chromosomes could be made of? Which are protein clumps. So DNA and protein. So during this time, when scientists were saying, well, obviously it's the chromosomes that are the inherited material, which is a true statement. The idea of, well, what's the inherited, like what is the molecule of inheritance? It was iffy or unknown if it was a DNA or if it was protein. So at this time it was unclear if DNA or protein was the genetic material. Cool. In 1928, and we'll fast forward, the experiment I want you to know at this time is Frederick Griffith. And his experiment was about the transformation of bacteria. And so let's go over that by drawing out the cute little picture of the mice. So what I'm going to get you to do is maybe draw this down real quick, something very simple, and then we'll move forward. Okay, so there were actually two strains of bacteria that Griffith was messing with. He was messing with one strain, the S strain. These cells were living S cells. And these cells, the living S cells, these bacteria are very pathogenic, which means that they can cause death. Well, that makes sense because obviously if you look at the bottom, you see that the mouse is dead. So a dead mouse because you were sticking in S bacteria, which were pathogenic. Now he also had a different type of bacteria called R bacteria. So right here are living R cells. Again, the R type of bacteria is essentially harmless. And so as you could guess, at the end the mouse is alive.
Yeah.
Works out perfectly. Right? Now in this case right here, in which the mouse is still alive, one would think, well, I've got to be giving it R cells for it to still be alive, but actually what he did is he used heat to kill the S bacteria, the S cells. So since the S cells were dead, right, that bacteria was dead, even if you injected it into the mouse was still alive. Do you understand why the mouse was still alive? No? Okay. Even though S represents a pathogenic bacteria cannot kill you or kill a mouse in this case because it itself is killed. The S bacteria is dead. Essentially, he boiled the bacteria before he put it in the mouse, and the bacteria was dead, so it didn't cause any harm. Cool.
Is this the same guy with like the crazy test tubes and stuff?
Now, this next experiment was actually probably the one that was the big one, like the most important one. He took heat-killed S cells, which we know from the previous experiment would do nothing to the mouse. and he mixed it living R cells. What would R normally do? If you just had living R cells, what would that do?
Nothing.
Yeah, the mouse would still be perfect, right? And if you had heat-killed S cells, as you did in the previous case, the mouse would be alive. So you're mixing two types of bacteria, one in which is dead and one that's alive, but in both of those independent cases, the mouse still survived. But you're noticing here that you have a dead mouse. So the no.
Dead mouse. It's a,
DJ.
yeah, it's a DJ.
MAE5.
That's not fine.
Yeah.
No,
Yes, you have.
no, I haven't.
You and your DJ buddy
Just
used to
like
go
you
down
hadn't heard of Justin Bieber.
Okay. So I think the important part to realize is that when he extracted blood from this mouse, he found that it contained bacteria, and this is the part to catch. Living S bacterial cells. The question would be, how could they be living? Because you had killed them, you injected them into the mouse, and it's like it killed the mouse, but it's like then life was brought back to the S cells, and that's actually not the way it was. What happened was the R cells absorbed, we now know this of course, they absorbed the DNA from the S cells. What did they do that for? When the heat-killed S cells enter the body, the DNA from their body, from those bacterial cells was released, because that's what happens when things die. Their cells pop open. That bacteria ended up in just the medium of blood that's in the mouse, and an event called transformation occurred that absorbed all that DNA. You know, like to transform something is to obviously change it. So in the transformation experiment, what he was noticing is that the good bacteria were being transformed into bad bacteria because they were absorbing bad DNA.
What happened to
This-
cells' own DNA?
No, their own DNA. They still had their own DNA, but they were absorbing the bad DNA of the S cells, and that DNA of the S cells gave them an advantage because obviously it's taking over the mouse's body. It's a parasite. So in the case of transformation, this is an event in which DNA is taken up.
Why did they look like S cells when you came up?
They did. Why did they?
Yeah.
Because R cells and S cells do look the same, except there's some extra DNA inside of an S cell. Once that S cell is dead, it releases its DNA because things that die are opened. That DNA that made it in an S cell is now in the R cell.
So that makes that R cell and S cell.
That's the event of transformation.
If I'm around Brad's DNA, like soaking in a tub of Brad's DNA, how come I don't take on any of Brad's traits?
You have to wear cut out, I'm sure.
Not really. I think we just color.
First off, like, this is how bacteria communicate. Like, this exchange of small DNA pieces is how they communicate. We don't have to communicate in that level. We can communicate just as we are now, without an exchange of DNA. So that's one thing. And two is that, like, you're correct. Like, how would they not? Why would there not be expression of that? Well, first off, I'm saying that there's not even an ability to transfer the DNA. And second off, you'd have to transfer it to each of the cells for it to have all of Brad or Sam DNA in the other individual, which is an unlikely event because you're talking about trillions of cells. So for a variety of reasons, and first and foremost, there's really no need to do it because this is their communication form for bacteria. We've got obviously other mechanisms. We're fine without needing to do an exchange of DNA. You know, this DNA gives you an advantage, right? Like this gave the R cells an advantage to survive and take over the mouse body, agreed? Do you want an advantage? Like, do you want like a hint for life? Stop at a stop sign. I've given you a helpful piece of advice, just like that DNA was a helpful unit. I've given you a helpful unit. No, seriously. It's a form of communication. So it's something to give you a better advantage and make you survive longer.
So did their DNA fuse?
Well, it's not that it's a fusion. It's just they keep their DNA in little packets called plasmids, basically.
Okay, yeah.
Question.
How do they know which DNA to pick up? Like, why don't they just pick up the DNA?
They take up any DNA. Yeah, but sadly, they wouldn't take up eukaryotic DNA of a mouse because the mouse is going to be releasing its normal DNA. It shouldn't be, and certainly even if it did, it's not going to match up with the DNA requirements of a prokaryote.
So if we have
Any other,
prokaryotic DNA,
specifically bacterial DNA, and honestly, if you want to know the truth, it can only take up certain types of that DNA. It's got to be.
lose their identity then over time. Yeah. Their original identity.
They're just different, what we call strains. So they're still E. coli, but they're strains of this or that. Like the influenza virus, yeah, well, that's a bad example, but there are plenty of strains like of strep throat, right? That's you're still streptococcus, but you have different little pieces of plasmid, little pieces of DNA that makes you a little bit different. Question?
So gaining that S DNA is an advantage to the R cells?
Yeah, because obviously now they're able to like take over and utilize more resources. Hence, that mouse is dead.
What they want to do is they want to kill the mouse?
Well, the overall goal is to proliferate and like max out what's called your carrying capacity, max out your resources. So a lot of times that does result in the death of the other creature. Usually, like in a quest for dominance, it's like things will die. Question.
Scenario: there's dead R cells and living S cells. Do the R cells turn into the, or do the S cells turn into R cells? Like, can you switch it?
No, because the S cells have an advantage for survival. Some of them would, but they would die off because they don't have that advantage. So, like evolution would go towards the way of just removal of all of the R's, and then the S would be just a value.
That advantage to kill the mouse in the first place?
What?
Is the S cells have the advantage to kill the mouse in the first place?
That's correct. Yeah.
Like if you had like one water bottle that's mostly full and one water bottle that's mostly empty, and you could choose between one, like you're gonna stick with the mostly full one, or you're gonna choose the mostly full one.
Well, I mean, it just depends. See, it depends. Like, imagine that you're in a world where there's no water. Well, then you obviously would choose the one that's the full one, correct?
Yeah.
But imagine that you're in a world where having water, like where water's explosive. Which would you choose then? The one that's almost empty, correct? So a lot of it's not your personal choice. If you think about it, it's the environment's choice. That's what evolution is. It's not choice of an individual. It's choice of an environment.
I was saying
This-
like which one is advantageous to.
It depends on the environment.
That's yeah. Saying our environment.
In our environment, in our life right now, a full water bottle is best.
Yeah.
If water became spontaneously going to explode, then it would become a detriment.
So that environment said that you
Even
more
we all
advantageous to have the DNA.
To have the sDNA, because that could destroy the creature. Okay, very good. Let's move forward. Okay, hello. Thanks for being here. Okay, not all at once. Let's keep going forward with stuff. I want you to know about, and you could write this down for Frederick Griffith, his experiment. He used the harmless R bacteria and the pathogenic S bacteria. You could mention just the Avery and McCartney-McLeod experiment just in your notes, which gave further proof that DNA was the genetic material. And then I want to jump into, right after that, a piece about Erwin Chargaff. So I'll let everyone catch up with this. I'm sorry, what? McCarty, I don't know, because I misspelled it incorrectly. McLeod, M-A-C-L-E-O-D, 1944. Just an experiment to further prove that this was the genetic material, or the DNA was the genetic material. Very good. The next thing I want to talk about is Erwin Chargaff and Chargaff's rules, which is a great, simple little AP test question. What he came up with is that this very, very basic and straightforward idea that the amount of A, the base A, adenine, would have to equate or be close to equal to the amount of T. And so subsequently the amount of G and the amount of C would have to be pretty close to equivalent. And we know A and T are together and then G and C are together. So A with T, G with C, that works out perfectly. So if you had a test question on the AP exam and they said that the cytosine amount was 15%, how much would the G percent be? Close to 15%, correct? So that would make 30% there, and these two have the 70% to share. So that means that the A percent would be 35%. Makes sense? Very like simple math. We skip forward a little bit and we talk about Rosalind Franklin, who received some credit for the structure of DNA, but not as much as probably she's deserving. She used X-ray crystallography to discover the shape of the DNA. Just like a freeze frame picture almost. Didn't come up with the exact structure, but came up with a very relative structure. Came up with this model of it being double helix, having a constant width. Good stuff? Yes? Okay. Now, again, I know you hate to draw stuff out, but, you know, it's all good. Okay, so what I want us to see is that in this experiment, which is the Hershey and Chase experiment, the year is 1952. And what they used was they used a couple of important items. And the first thing I want to go over is this concept of a bacteriophage. What is a bacteriophage? Does anyone know? It what? Okay. It's a virus that destroys or invades bacteria. Good.
What's the year?
What? 1952. Cool. What they had were two setups that were virtually identical, but in this top case they used an isotope of sulfur, so they used radioactive sulfur or an isotope of sulfur. In the bottom case, they used not a radioactive sulfur, but they used a radioactive phosphorus. And from the reading, does anyone know why that they would choose to use that?
They said that it like attached to it, phosphates attached to the DNA, and then sulfur was with the protein.
Exactly. The sulfur is an important element of protein, whereas the phosphorus is an important element of the DNA. You can see here that this was kind of the definitive experiment that guaranteed that DNA was going to be the genetic material. Good? Now, in either which case, you see that the drawing is essentially the same. You've got a bacteriophage that's infecting a bacteria. Correct? But what they see in the top part is you'll notice that there's the protein that makes up the virus. See, they incorporated this radioactive sulfur into the viral protein. But see, the protein is what does not enter into the cell. So that means at the end, when you kind of divide everything up, you still see your protein inside of the virus, and that was not the transformation agent. So this proves that protein is not the transformation agent. But we did see that when you use the radioactive phosphorus, which was in the DNA that was inserted into the bacteria was this isotope of the DNA of the phosphorus. and we examined this in the end. So really what this comes down to is the simple fact that when they checked the bacteria cell at the end, the only radioactive isotope that they saw was not sulfur but phosphorus. Makes sense? See that the sulfur isotope never ever, and you'll see from the no purple inside of these cells, the sulfur, the protein, never went inside of the bacteria cell. It can't be the transformation agent because it was never transforming the bacteria. But however, this phosphorus, which of course is a part of DNA, so the DNA is now labeled. The DNA was going inside of that bacteria and changing it up. So this is like verification that the DNA has to be the material transformation. Right? I hope we get that. The radioactive isotope is just a tracing mechanism, and always has been. It's when they give you an MRI, and we're doing like fossil recording or fossil dating. It's always like important for tracing stuff. Right? So the Hershey and Chase experiment started with the Griffith experiment, Avery McCarty experiment between the two, and then of course Hershey and Chase at the very, very end. Now, the benefit of this is setting our stage up for the mid-1950s and the discoveries of Watson and Crick, in which the structure of DNA is discovered.
Structure of DNA?
The structure of DNA. The overall structure of how DNA is put together. And as we see tomorrow, their paper was about the structure of DNA, which is a great, I guess, idea or topic. What's more valuable, though, I think, is that at the end of their paper, they just didn't say, well, you know, this is the conclusion of the structure. They said, not only do we have the structure and we're pretty confident on this is what DNA looks like, but they came up with a possible mechanism of replication. And about 3 years after they had hypothesized on the method of DNA replication, it was actually proven to be true. So, I mean, that's really like why they're so valuable, is because they were able to guess exactly how the DNA was being copied as well. Cool. So in our last 5 minutes, real quick, I want to go over this, which is the structure of DNA. Again, this is all in your book, but I'll just go over the parts that I think are important. You'll notice that this piece right up here, I want to talk about just this one unit, is a nucleotide made of 3 important pieces. You have the base off to the right, you have the sugar, and of course the phosphate group, right, Adam? Amazing. So that's 3 parts that make up a nucleotide: a sugar, a phosphate, and a what? Base, right, a nitrogenous base. And there's 4 different bases for DNA. This is the base of thymine, this is that of adenine, there's cytosine, and there's guanine right there. If you remember from back in the day, I said we can divide these up into 2 groups, the purines as opposed to the pyrimidines. The purines were which 2? A and G. And that meant that the pyrimidines were T, C, and U. TCU versus AG. Awesome. Ag.
Wait, what is the base again?
The base are these pieces over here.
Yeah.
A, T, G, and C. Right? And you know A was always with T and G is with C. Which means if you think about it, when you combine these, when you set the other one over here, and you can see that in this bottom picture, hopefully you're with me still. You'll notice that you always have A with T and G with C. Right? So you always have a purine bound to a pyrimidine. Right? You'll notice that the binding is actually these little dots. Do you know what type of bond that is? So it's a hydrogen bond, which means it's not as strong as a covalent. It's fairly strong, though. It can be broken. Still, it can be broken. But yes, there are tons of them, which is why the structure will stay as it is. Great. It's also valuable that you be able to count the carbons on deoxyribose. And this is deoxyribose because I see it position 2. There's no hydroxyl group, no OH, which means it's going to be deoxyribose. Ribose itself is a 5-carbon sugar. You have to be able to count out the 5 carbons of that sugar. There's carbon 1, 2, 3, 4, and 5. Right? That means that the phosphate group is connected to carbon 5, whereas the base is connected to carbon 1. Right? Good? We got this? So carbon 1, the base, carbon 5, the phosphate. Number your carbons 1 through 3. If it's deoxyribonucleic acid, DNA, no hydroxyl on position 2. And then you'll notice, like at the very bottom, and this I think is probably the most important piece of today, I could label this as being the 5' carbon. There's your 3', 5 and 3, 5. See, I'm kind of tracing this all the way down. So I would label this end as the 5' and this is the 3' end, going that direction, because you always go from the 5 to the 3. Right? Now you'll notice that this strand, though, is like the direct opposite. So it is parallel, but it is running in the opposite direction. It is anti-parallel. Right? That means that this carbon right here is going to be carbon 3. This carbon right there is going to be carbon 5. You always go in a 5 to 3 direction. So in tomorrow's stuff, we're going to go over explicitly, or we will start, how this system could copy. Don't forget about bringing the M&Ms for the lab on Friday. Right? And that's about it.